for the invitation, and I'm happy to see that so many of you took the time. I mean, it's the lunch break, so maybe you're eating at the same time. I don't care. My own background is in, I cannot change the slide. I think I'll just stop sharing and then try sharing again. Maybe that's going to work then.
Yes, go ahead.
That's really strange.
Still nothing?
No.
Are you sharing your screen or the program, the PowerPoint?
I'm sharing my screen. Let me see. Share from the beginning. Now it works. Okay,
perfect.
So I'll tell you a little bit about what we do in our Center of Excellence that we have named IDUL. And here you can see that we believe in bringing science to life, and I'll tell you what we think when we say that. And then our ambition is to do good science and to grow better scientists, but also to create new businesses. And below you can see the values that we believe in and what we strive to behave like. Kind of how we were established happened 6 years ago when we more or less at the same time, we got two grants, more or less simultaneously. One to do what we had been doing for many years in nano and micro sensors. So that's what you see on the right-hand side, sponsored by a private foundation. And then we actually were allowed to do something completely new for us, which was drug delivery, using some of the technologies that we have in micro and nano fabrication. And since it was two different grants, we were kind of asked if we could merge them, because then the administration would be much easier. And I thought in the beginning that's an awkward thing, but it really showed that you can have a happy marriage out of this, because there's so much in drug delivery where our sensors can be applied. And also you can see sometimes that there's a link between the needs for diagnostics and delivery, for example. So it has really, created a lot of new ideas and new research and new directions by actually merging the two activities into one. And by the way, my own background is originally in physics, so I've also kind of traveled a long way to now also be working with drug delivery. In terms of bringing science to life, it's our ambition to implement what we call top-down micro and nanotechnology discoveries to life science challenges. But it also describes that we really like to translate some of our activities into more practical applications. So I have a few examples of that as well. So this is us when we're all gathered. So we were so fortunate to get out of the Corona shadow in May and go to a nice location for 2 days north of Copenhagen, very close to Sweden. And you can see us here. What you cannot see is that we range all the way from computer science to pharmaceutical sciences. It's a collaboration between University of Copenhagen, SSI, so this is where we do vaccine development in Copenhagen, and then the Technical University where I'm based. And maybe you know some of the senior people who have been involved, like Annette Müllers, Hanne Mørk, Thomas Rates, and Jukka Rantanen, as some of the participants from University of Copenhagen. If you're interested, you can also follow us on social media like LinkedIn and Facebook and Twitter. So where do we come from? So I come from the Technical University. I started my own research group in what's called nanomechanical sensors. We have done a lot of what's called centrifugal microfluidics, doing liquid handling on disks. We love to hack devices that have already been disrupted, like how can you maybe repurpose a Blu-ray player? And then we build huge optical setups for doing what's called Raman-based analysis. And we do a lot of nano and micro structuring, for example, these surface-enhanced Raman scattering substrates, polymer structures for drug delivery. And if you haven't seen it before, then the last picture here is an image from our clean room facilities. So that's where we do a lot of our devices working in these ultra clean facilities because we're structuring materials down to the nanometer scale. So we need to work in a dust-free environment. So for today, I've chosen 4 stories that I would like to share with you that kind of connect some of our sensor activities and drug delivery activities. And the first one relates to how we can characterize very small amount of drugs. Move on to an example of therapeutic drug monitoring, then a small story on how we can grow cells on disks, and then finally some of our activities in oral drug delivery. So first, the characterization of small amounts of drugs, and that's something that I was not aware of, but our collaborators at University of Copenhagen, they're really aware of how important it is in a very early formulation stage to know how drugs behave, for example, when it comes to temperature changes or humidity changes. And to do some of these more fundamental studies, we were kind of using sensors that we have been developing and using for many, many years, but now with a new application that our collaborators could bring in. What you see here, it's basically a guitar string, but it's super tiny. It's made in silicon nitride, and the support structure is silicon. You can resonate it, you can shake it, so it's going to resonate at a very defined resonance frequency. And if the mass is changed or if temperature changes, it's going to detune, it's going to change its resonance frequency. Just like a guitar would also detune if you took it to a room with a different temperature, because the guitar string and the bass, they're going to expand differently if you take them to a new temperature. So that's what we are using to create an ultra-sensitive temperature sensor. And the way we're going to measure is how much this little string is going to detune. How much is it going to change its resonance frequency? So Kalle here, he came along from University of Copenhagen with a background in pharmaceutical sciences. And he also had with him a lot of different drugs and also some new formulations. And he really wanted to see the phase transitions in these new materials. And what he did then was to take some of these resonators that you see here from above, and he would drizzle a little bit of the drug onto each of them, put it into a little temperature environment, and then ramp up the temperature and down again, and then at the same time measure how much these strings are detuning as a function of temperature. And he could actually see phase transitions and subphase transition, TG transitions in less than 1 nanogram of material, and also in materials that hadn't really been characterized before. So it was really, really interesting technology for analyzing small amounts of drugs here. Then Peter Okeo, he came along also from University of Copenhagen. And together with Yuka, he's very, very interested in studying how water is getting into drug crystals and leaving the drug crystals again. In some cases, some water channels are created within these drug crystals. And in the beginning, Peter, he really struggled to see if he could place some of his crystals of drug on top of our guitar strings or resonators, as you like. And in the end, Peter Okeo and his co-supervisor, another guy called Peter, they figured out instead of trying to place drug crystals on top of our clean room fabricated resonators, why not just use the drug crystal itself as a little resonator, as a little guitar string or cantilever. So that's what you see here in the bottom. You see one drug crystal that we then start to shake, and we measure how the resonance frequency is changing, again as a function of temperature. And what we could follow then in these drug crystals is, first of all, we just see them from above, and this is kind of a time scale, same crystal over time as we ramp up the temperature. When all the water has left the crystal, it's going to turn more black. So we can see visually something has happened. And at the same time, the way we measure the resonance frequency actually also shows clearly that something has changed. The mechanical performance has really changed a lot within this drug crystal. And this then gives us new fundamental insight in how water is interacting with these drugs. So it's another example of where we kind of, in a very simple way, use some of our sensor technologies for characterizing drugs. The last example in terms of sensing on drugs is this one. It's our Raman activity. So it's really very much built on Oleksi's experience in building new optical setups. And then here it's together with Chiara and Roman. So we have now a home-built system that can do 3D Raman imaging of, for example, drugs or formulations. This is a case where we have one of our microcontainers that we have made for carrying drug inside. And with the Raman chemical mapping, we can actually visualize where do we have drug, then it's colored red in this case, where do we have a carrier polymer, that's green, and where do we have the surrounding container, that's yellow in this case. And the whole device is around 300 microns in diameter. So kind of using the sensor guys that we have in our group, we can actually characterize some of the things that we're developing within drug delivery in a rather unique manner, I would say. What Oleksi and co-workers have then also done is to use, instead of normal Raman imaging, you can use polarized light. And this way you can get entirely new information. Normally, if you just map the top of a drug tablet in this case, you could see where do I have a drug, so this is red. Where do I have a carrying polymer, that's green. And then the blue parts, that's some background fluorescence. If you then turn on your fluorescence, your polarized light, you will see that the red region here, that's the drug region, it actually contains a lot of small domains. And that's because we can now also map the orientation within the drug part. So we can actually now do imaging that you would normally need to do at a synchrotron, just using a Raman setup. So that's really cool, a nice tool, and something that Oleksi has then also spun out a company on, which is called Light Novo, where this technology is being developed further and also sold for people who are interested in that. So that was my first story. The second story is on doing diagnostics and therapeutic drug monitoring using just 1 drop of blood. So the first case here is the current situation where we have coronavirus, unfortunately, and we are being vaccinated. Still, there is really a need for monitoring the effect of the vaccination, and that you can do by monitoring the amount of antibodies that you have in your blood. And for that, you need cheap technology if really you want to spread this out for broad use. And one of the things you can do is actually to repurpose your Blu-ray player. Inside a Blu-ray player, you'll have the disc that spins really fast. And on top of that, you have optics. Normally, you would have 3 lasers inside that all have a spot size of less than 1 micron. And they're already optimized for perfection. So you can use them to build a lot of new instrumentation. And that's what we do with the help of my colleague, Edmund. This is an example from our lab 6 years ago, where you see a disc with the infrared channels. You can insert 1 drop of blood, and you can spin the disc. And this way you can centrifuge the blood sample and just separate out the blood serum and transfer that to a chamber where you're preloaded magnetic.
Sorry to interrupt, Anja. It's hard to hear you.
All the time.
You had some disturbances.
I think
maybe you can move.
Is it better now?
No. Do you have another mic you can use?
Is it when the movie was playing?
Now it's much better.
It's maybe the bandwidth with the movie could be.
Okay.
But otherwise, let me know again. I might be able to find a professional mic. Good. So this way of actually centrifuging the blood sample and then actually measuring inside the disc, we can measure agglutination assays in this way with a blue laser from a Blu-ray player. It's a company now called Blue Sense Diagnostics, and you can see our former PhDs and postdocs here who founded this company. And there are now around 60 people in Copenhagen and in Taiwan. So now we thought, okay, maybe we can do something different now using partly some of the same technology, at least the disc. So we are working on now is called therapeutic drug monitoring. where we go in and see if we can help in the treatment of patients with cancer, specifically children with leukemia. Because the way they are treated requires therapeutic drug monitoring. In Denmark, as a child with leukemia, you're getting very high dose of a drug called MTX. And if you are above a certain concentration, you're going to have severe side effects, and they need to administer an antidote. So by regulation, you need to monitor these patients very closely, and there are very few hospitals in Europe that can actually do this. So we would really like to see if we can spread out this treatment by offering a way of doing the therapeutic drug monitoring. And we do this in collaboration with a strong key opinion leader. He's the head of the pediatric oncology unit at Rigshospitalet in Copenhagen, Kej Mikkelov. And he is actually the one who approached us and told us how important it is to do this type of monitoring. So our first test case here is the drug MTX. And we detect it by Raman scattering because it's a small molecule and it's really good for Raman scattering. But it's not enough to do normal Raman imaging. Here we don't have the sensitivity that we would need. So instead we use surface-enhanced Raman scattering. It's a technology that's been known since the 70s. So what you need to do is to somehow place your molecule, your MTX, on a rough metallic surface. Because then what will happen is that it will kind of force light to scatter in these molecules. So the principle is you send light in one wavelength, it's going to scatter in the molecules. At least some of the light is going to scatter, and it's going to come back with a slightly shift in wavelength. And it's this shift in wavelength, what comes in and what comes out, that's going to give you a chemical fingerprint of what you have in your sample. So to make this nano rough surface where we can place the MTX on top, we etch into silicon. We call it nano grass, and then we cover it with gold or silver. And then in the end, it looks like this. So you can see it's really rough nano grass, and you can do it on huge surfaces for your measurements there. So we are not normally working with huge surfaces. We cut these small pieces out, maybe less than a centimeter by a centimeter, just a few millimeters. And then we can store them, use them ourselves, or send them to collaborators. And we also have a little startup company called Silmeco, where researchers, they can buy their own substrates and use them for developing their own applications. So to do the therapeutic drug monitoring part, we use that we now know how to make these rough metallic surfaces. We also know that these surfaces can be applied as an electrode. So if you know the charge of the molecules you would like to detect, you can actually apply the opposite charge and then attract the molecules to your surface. In this way, we know we can increase sensitivity. And we also know how to integrate these search chips onto a spinning disk, so we can combine it with the blood sample treatment I showed you before. And we also know that we can wirelessly communicate with all the electronics on top of such a spinning disk. So we kind of have all the ingredients in place for doing all of this. Just to show you what you can do if you play one of these nanotextured surfaces into a spinning disk, for example. This is not blood. In this case, it's milk, and we have also tried with urine and with blood. But you can see that only half of this nanopillar substrate is actually placed or dipped into the milk solution. The disk is spinning, but we are illuminating with stroboscopic light. So it looks like it's standing still, but we are just providing the solution, and it's being drawn up through the nanopillars, because the nanopillars, they work as a filtered paper. So that kind of the solution is going to move up. You can maybe see that it's changing, it's becoming darker and darker, but only the very tiny molecules will be able to travel through the nanopillars. So the nanopillars, they function as a filter. And on top of that, they also work like a spatial separation of molecules, because depending on the affinity of the small molecules in your sample, they're going to travel a small distance or a longer distance. So it works in the same manner as thin film liquid chromatography, just on a very small sample surface. And this is just for milk case, where you can see it looks like it's completely clean in the top part here. And afterwards, when you dry out the sample, it's very dirty in the bottom. And if you then try to do the Raman imaging in the two regions, you see a clear signal. In this case, it's melamine and milk. And there's nothing down here in the contaminated region because you have too much stuff in the milk sample. So it really shows that we can actually reduce the background signal just measuring on the kind of dry region up here. And what we also then found was this thin film liquid chromatography effect. This is for a urine sample where we can actually spatially separate out different drugs and components in a urine sample by dipping only half of the search chip into the urine in this case. So now we're actually doing the same with blood samples. So we work with the MTX in blood. And in the beginning, this is a paper that Yaman just got out. We were simply just dipping these nanopillar substrates into blood serum spiked with MTX, and then measuring on the dry part of these Raman scattering pillars up here. And it works. We can really clearly see the peaks from the MTX molecules, even in a rather complex background. We are now working on building a little instrument, a portable unit that can be standing next to the bed. And then we're doing a lot of calibration curves. And we're also receiving first patient samples from Rigshospitalet, where we get a sample before the children are treated with MTX and then one after or several after treatment, so that we can actually do our own little calibration curve using the sample from before the treatment starts. In this case, it's from two children undergoing MTX treatment. And then we're comparing to the standard ELISA assay that they're running at the big robotic machines at the hospital. And you can see in one patient, we're very close to the concentration they're measuring. And in the other, we are kind of something like 50% off. And this is really related to the way we need to pre-treat the blood sample, because this is where we really need to improve. Work in progress. This is a 3-year project funded by something called the Bioinnovation Institute in Denmark, funded by the Novo Nordisk Foundation. So that was my second story. My third very short story is on how we can actually grow cells and how sometimes our ideas happen more or less by coincidence, because there are actually a lot of my colleagues also working on different sorts of in vitro systems. And it's not really something that we're planning to get into as such. But it happened that Stine, you can see down here, she was doing a treatment of bacterial biofilms using our microcontainers loaded with antibiotics. And to test how well these were performing, we've got in contact with my colleagues here, Søren Molin and Helle Johansen. Helle is also a medical doctor at Rigshospitalet, and Søren Molin is really one of the key experts in Denmark in bacterial biofilms. And they have their own little in vitro systems where they grow a bacteria called Pseudomonas aeruginosa in small lab-on-a-chip devices. And then we saw their setup, and you can see it's here. So it's a lot of polymer chips with channels, a lot of tubing, bubble traps, pumps, and you need a lot of growth media to actually provide growth for all these bacteria, because also you have so much dead volume. I mean, so many tubes. So then Laura, she was joining Stine, and Laura has been working a lot with centrifugal microfluidics and doing sample pre-treatment on discs. And she thought, I think I can do everything on a single disc. And this is what you then see here. So it's an inner reservoir where you load growth media for up to a week of cell growth. You inoculate your cells here, and you grow them in the central chamber here, and then you have a waste reservoir around. Then you spin the disc slowly, and that spinning is going to press out the liquid. So there's a constant flow of nutrition to your cells. You don't need a pump, and you won't have any issues with the bubbles. And you can place it directly in an incubator, and the only thing you need is a little motor that can spin the disc. If you need to do more cultures, you can stack the discs on top of each other, and you can even monitor the growth over time, as I'll show you here, because then Edwin got involved, and he mounted a little spy camera on top of the disc and a little lens, so that we now have a microscope that spins along with the disc inside the incubator, and it transmits wirelessly the signal to outside the incubator. And this way we can monitor over time how these cells are growing, and you can do these cytotoxicity assays if you like. And then we can actually treat the data with a little spin-up company that also came out of our center called Celery, where they use AI to actually recognize the cells and count them over time. So that will bring me to my last story, which is on oral drug delivery. And here we started with these micro devices, and you can see them here. They kind of have the size of sugar grains. Our ambition here is to provide our little, maybe more technology-oriented solution to kind of getting a little bit away from all the needle injections. And as I think you know in this audience is that it's really preferred to do oral delivery, but a lot of times it's difficult because the drugs are either being degraded as they travel through the stomach, or they have a very hard time getting across the intestinal barrier, or a combination of both issues. So we thought, okay, maybe instead of trying to make new formulations, because that's certainly not our background or competence, maybe we can use all our knowledge in micro and nano fabrication and see if we can come up with some solutions that might make it easier for some of these drugs to get where they need to get. So we started out making these microcontainers. They are 300 microns in diameter, made in a polymer. You can fill them with drugs, and you can put some sort of a lid on top. It could be a pH sensitive lid, and you can also play around with nucleoadhesive coatings. And then ideally, they should orient themselves right where we want them to be. And they only release in one direction. So it's really a unidirectional release, and everything should be fine. So that's kind of the dream scenario. You could think of some of the benefits. First of all, we know how to fabricate devices. We can do it in a very controlled manner. So we have complete control of size and shape. To some degree, we can control where we're going to release. pH-sensitive lids, for example. We have pretty high loading capacity. We can fill everything inside with drug. The drug is only going to come out in one direction, and we can combine a lot of different drugs if need be. And we have also seen that putting amorphous drugs inside these small cavities actually stabilizes the drugs quite a lot. And probably it's because you're reducing the risk of nucleation when you just put this drug into small compartments. Since we just started with the small, simple containers, we have ventured into a lot of different directions, sizes, shapes, materials, depending on what issues we're facing in a certain application. I won't go through all of these different devices. I'm going to talk you through the very simple one and then show you some examples in the end. But I think in this way, you can get an idea of how we're working with this. In terms of making these small polymer containers, we do what's called lithography. So initially, we started using polymers that are photosensitive, so you can structure them by light. This is just the process. You can see a polymer on a silicon carrier wafer. You shine with UV light through a photomask. That's going to define the bottom of a container. And then you spin on a thicker layer seen from the side of the same polymer, and you illuminate again, and that's going to define the side walls of a container. And then you can develop the whole thing, and you're going to have these small containers sitting on a substrate that you can release them from in the end. This is what they look like when they've been fabricated. And in each of these small squares here, you have 625 containers. And that's normally what we use, for example, in one rat experiment. The volume in these containers is around 10 nanoliters. And if you really want to fill them with drug, and we have many ways of doing that now, but initially when we had the first containers, we were really thinking, oh, okay, then how are we going to fill it with drug? But over the years, we have developed a lot of different tools and technologies for doing this. I'm just going to show you one that's been developed by Kashit. She's coating everything outside the containers with a PDMS, a polymer. It's not going to go inside the containers, as you can see here down in the images. So then you can take the whole thing and put it upside down and just stamp it hard into a powder drug formulation, for example. And this way, you get powder drug inside, and you can remove this polymer in between in the end, so you don't have any residual drug between your containers. This is what it looks like when it's been coated with a powdered rock or loaded with a powdered rock. And in this case, we have around 4 micrograms of material in each of these devices. To put on a pH-sensitive lids, you can buy a lot of these coatings or polymers that have already been developed for coating tablets. So we use the same ones. And then you can put them on top using, for example, a spray coating setup. But there are other techniques you could use here. So this is an example of what a little container looks like when it's been coated with a pH-sensitive lid. And then normally before we go into animal studies, we do some in vitro characterization using, for example, media that look very much like the stomach fluid and the intestinal fluid. And then we simply check how much release do we have in these two regimes. Does it behave as it should? And we normally also inspect these devices afterwards in scanning in a electron microscope just to see if the lid is still there and the drug has actually come out. To do the animal experiments, we release the containers from the carrying substrate, and we just fill it into some of these gelatin capsules, depending on what type of animals we're going to work with. Here you can see them filled into one of these gelatin capsules, and then we're ready for in vivo experiments. And we do oral dosing with the device so that the animals are not going to chew on the gelatin capsules. So we really would like to have complete control of how many devices we have delivered. And then we normally take blood samples over time. We work with controls all the time. For our first experiments, they look like this, so that we would code or we would load small molecule drugs into our containers and load with a pH-sensitive lid. And we would actually have the same amount of powdered drug in the big gelatin capsule and the same coating on the big capsule. And then we would take blood samples for the first 24 hours and working with groups of rats. The first studies we did, that's done by Lina and then later Clara and Fabio. So it was with furosemide and ketoprophene, the first things we did. And this is one example. This is for the furosemide studies, where we could see that if we use the microcontainers, we have more or less the same peak value as we would have for the big urinated coated capsules, but that the release is much prolonged for the microcontainers, and we also have a much higher overall oral bioavailability. You can also say that we should have extended this study, but we didn't really think that we would have this much material still in the samples after 24 hours. Just looking at the tissue afterwards, we could actually see that some of these containers, they embed very deeply in the mucus layer in the intestine, and also some of them very close to the epithelial wall. And this could be some of the explanation why we see this very prolonged release. In terms of what we're doing now, we are working together with DTU Food on delivery of probiotics. We work on delivery of antibiotics. We have activities in gut sampling, and in insulin delivery, and also in oral vaccine delivery. And I'll show you a few of the things now that we are working on. But before that, I'll just say that we do have some challenges ahead of us. First of all, especially for our vaccine studies, we've seen that we need to increase the residence time, especially when we work with mice and rats. We're not working with humans, and we would really like to increase the interaction between our devices and what we've loaded inside and the animals. And in a mouse, for example, our containers, they are all the way out again within 3 hours. So that's something we're working on now, how to increase that. Another issue is that we would like to work with materials that are biodegradable. So far, the devices I showed you, they were made in an epoxy-based material, and they are not going to degrade. And I'm not even sure they would be approved for human use in the end. So that's something we're also working on. In terms of increasing residence time, we've tried to play around with making devices that had some sort of arms or fingers sticking out so that they would attach better to the mucus layer, for example. And to do that in a high throughput way or in a very where you can easily change the design. We've been using 3D printing. And it's Lucas who first had to optimize how can we actually 3D print small structures that we can then release in a safe manner. And when he had that in place, he could start making some of these devices, as you can see here. And the one all the way to the right has been inspired by a bacteriophage, the look of it. And not that you should go through all the graphs. I know this is a really busy slide. It's taken from Lucas's paper. But he designed this little slide here where he can mount a piece of tissue from a pig intestine, and then he would place all these different devices, either with the head down or head up, and then he would flush through a medium here, and then he would just monitor when are the devices coming off. And he could then, by this, see that if the arms are sticking into the tissue, they stay much better than if it's the back end sticking. I guess that makes sense. And also that some of the designs, like the phage design here, stays much better than some of the others. All of this looks good when you do it, these type of tests. If you try to repeat it in animals, in rats, for example, we see only a tiny difference. So here the solution has to be even more extreme if you really would like to increase the residence time. To actually see what we're doing, we're now doing much more X-ray CT scanning, and that's the work of Prof here. And that's also now adding to our understanding on how these devices travel through the body and where they stay. And what we need to do here is to either make the containers with embedded contrast agent in the polymer itself, or we simply load the contrast agent instead of drugs into our micro devices. And this way we can actually now track how the micro devices are traveling through small rodents. And in terms of the biodegradability, we have also, thanks to the work of Samina and Stefan Keller, we have been able to transfer our fabrication into biodegradable materials, like, for example, PCL and PLGA. It means that you need to rework your entire fabrication process, but this we have done. A few examples in my last minutes here. So for the delivery of antibiotics, we worked with Søren Molin and Helle. I showed you those faces before. So Stine, she loaded antibiotics into our containers and coated with lids on top. Also played around with the mucoadhesive lids here. And she could actually see that compared to a bolus dose of antibiotics, or flushing through the same amount of antibiotics in a diffusion model, our containers, they kill 3 times as much biomass. But this is in an in vitro system. So at least here it looks really promising. And we can see that the containers really embed in this mucus-like biofilm that we have generated with Pseudomonas aeruginosa. In terms of our second application, that's insulin delivery, and it's something we wrote into the proposal because we know it's really challenging, and we also have a big industry here in Denmark that's interested in this. But it is really difficult, and I guess you know that insulin doesn't want to go through the intestinal wall. So with our collaborators at University of Copenhagen, we've been working with these permeation enhancers that can open the tight junctions so that insulin can actually go through. And we thought then that our microcontainers, they could serve this purpose of co-localizing the permeation enhancer and the insulin, and we could see, what's the effect of co-localization. And to do that, Jakob from University of Copenhagen, he made this Cargo 2 cell mod, and he placed the containers just on top. And then he would measure if he gave permeation enhancer and insulin together with the containers, but the containers themselves are insulin, nothing is actually getting across the Cargo 2 cell. So the containers themselves are not really rupturing the cells. If you then co-localize the insulin and the permeation enhancer, you can see just a little less than 20% is getting across the cell layer. And if you put half of the 625 containers with the insulin and the other half with the permeation enhancer, you do get something across, but not that much. So co-localization is really important. Then you can play around with the separation. So you're just, slowly lifting up the containers further and further away from the CARCO2 cell layer, and you're monitoring, does it have an effect on how much insulin you get transported through? And sadly enough, if you're just 1 millimeter away, basically you get nothing across. And it kind of shows us also that it's really important to be more or less really, really close to the intestinal wall if this is ever going to work. And on top of that, Jakob tried twice to do a rat experiment, dosing them with microcontainers with this formulation inside, and he couldn't see anything in the blood of the rats. So we had to rethink. And what we're now working on with Mehdi and Carmen and Lasse is to make new devices. We call them foil devices. So these are devices that will unfold when they get out of the gelatin capsule in the intestine. And then they're going to align and expand a bit, towards the intestinal wall. And maybe you can see here through the intestinal wall that we do have container geometries inside the foil. And this is where we are holding the formulation. And this is Jakob's data showing that the gelatin capsule, if the gelatin capsule is uncoated, it's going to dissolve pretty fast, and then the foil can unfold and release whatever it has inside. We did have first data published, and that's together with Jakob and Hanne Mørk and Thomas Rades and Annette Müllers from University of Copenhagen, that we actually, using these foils in rats, could see that we do deliver a small amount of insulin now. For the first time, we can see that using the foil devices. The bioavailability is below 1%, but it's still, our first proof of concept. Finally, you can think of repurposing some of our devices to do the opposite of what we normally do. Normally we deliver, but maybe instead we could take something out of the gut. And especially from the upper part of our gut, the small intestine, is very hard to sample. Further down, maybe you can work with a fecal sample. So this started with when Sarvesh joined us. He has a background in micromotors, and together with Fatima, she has a background in tissue engineering, and she's really good in growing cells. So here, Sarvesh has repurposed one of our microcontainers so that it has a catalyst inside, so it's going to propel forward because it's going to generate bubbles when it's exposed to liquid. And it also has precursors for hydrogel formation inside. So it's going to start a polymerization of a tail that comes out here. It's a hydrogel. And as the tail grows, it's going to embed whatever it meets on its way. So inside it, if it travels around in a cell culture, it's going to capture cells. So this was the first proof of concept in vitro. And then Lou and Lina and Sarvesh continued with rat experiment, where they gave some of these devices to a rat, and then they would look at, when it then came out again, look at what did the polymer actually entrap. It has a lot of bacteria inside it, and the bacteria resembles the bacteria we would find if we would open up the rat afterwards and look what type of bacteria are actually in here. So it is kind of an idea for a sampling device that will go through your intestine and just embed whatever it needs on its way. So that's for sure something we're going to continue, maybe more with mechanical devices in the future. So that brings me to the end. It's the end of my 4 stories. I would like to thank our sponsors that have made all of this possible. If you're interested, we have a summer school every year in August where you can work for 2 weeks with us, either hands-on, it is, in drug delivery and sensing, and we have some really cool speakers as well. And just really, I would like to thank this awesome group of people who are making it so much fun to work with this. And yeah, thanks for all the work we do. And thanks for you for listening.